Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is September 27, 2025. This video is on a press release by the biotech company Unicure, released on September 24, 2025, regarding some exciting results regarding gene therapy in Huntington's disease. Huntington's disease is a rare inherited neurodegenerative disorder caused by cytosine adenosine guanine CAG repeat expansion in Huntington gene, leads to motor dysfunction, behavioral abnormalities, cognitive decline, no approved therapies to slow progression. The study investigators conducted this pivotal phase 1, phase 2 gene therapy trial of AMT-130, that is adeno-associated viral vector serotype 5 carrying microRNA targeting Huntington gene mRNA. The study cohort comprised of 29 patients with early manifest Huntington's disease. 17 were given high dose and 12 were given low dose. 12 patients per dose group completed 36-month follow-up. They were compared to an external control from Enroll-HD, Huntington's disease natural history dataset. 940 were matched with a high dose, 626 were matched with a low dose. Delivery, single administration via MRI guided convection enhanced stereotactic neurosurgical infusion into the striatum. The striatum is the caudate nucleus and the putamen. Duration 36-month follow-up, extended 5-year monitoring planned. Statistical analysis, propensity score matched comparisons under pre-specified statistical analysis plan submitted to the US FDA. What did they find? The primary endpoint was met, 75% slowing of the disease progression by the composite Unified Huntington's Disease Rating Scale, cUHDRS, at 36 months versus control, p equals 0.003. Secondary endpoint, 60% slowing of decline in total functional capacity, p-value 0.033. Cognitive outcomes, 113% slowing in Stroop word reading test, SWRT, which measures executive function, p-value 0.0021. 88% slowing in symbol digit modalities test, SDMT, which is psychomotor speed, p-value equals 0.057. Motor outcome, 59% slowing in the total motor score, p-value equals 0.1741. Biomarker, CSF neurofilament light chain, a marker of axonal degeneration, was reduced by 8.2% from baseline. Safety, generally well tolerated. Most adverse events were related to the neurosurgical procedure and resolved. Limitations of the study, small cohort, 29 individuals. External cohort, Enroll-HD, may introduce selection bias. Durability beyond 36 months requires ongoing follow-up. Key takeaways, AMT-130 demonstrates statistically significant disease-modifying effects in Huntington's disease. Evidence of dose-response benefit. High dose shows stronger effect than low dose. It's a potential landmark advance in Huntington's disease therapy. The biologics license application submission to FDA is planned for 2026. Key eponyms: George Huntington's chorea, the original name of the disease, 1872. Westphal variant, juvenile onset Huntington's disease with rigidity and bradykinesia, described by Carl Westphal. Vox athetosis, Oscar Vox's work on hyperkinetic syndromes influencing Huntington's disease classification. The Wechsler's cohort, landmark Venezuelan kindred study by Nancy Wechsler, enabled gene mapping. Schulzen-Fawn scale, functional capacity rating applied in Huntington's disease clinical research. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom.